we're almost there. We've captured the light energy. We now need to send it into the light-independent reactions to fix carbon. So we will again talk about carbon fixation, and to do that, we have to talk about the Calvin cycle, and then we will talk about how photosynthesis works as an energy cycle for all living organisms, and we'll talk about some different types of photosynthesis that occur. So here's an important enzyme, Rubisco. That's the shortened version of it. So this enzyme is kind of estimated to be the most abundant enzyme on Earth. It's very important in this light-independent reactions. So we saw in the last section the light-dependent reactions capturing that light energy. In the previous section, we saw the photosystems harvesting that light energy. They used it to make a hydrogen ion gradient that went through ATP synthase. We produced ATP. We also captured high energy electrons in the form of NADPH. ATP and NADPH are going to be fed into the second part, right? Those are the light dependent. We are now going to look at the light independent reactions. The light independent reactions are also called the Calvin cycle for the person who first described it. The critical bit here is we're taking CO2 and fixing it, carbon fixation, using energy to stick carbon from CO2 together to create organic molecules, which we will call sugars. These ATP and NADPH get used up real quick. We're talking millionths of a second. They are in and out of this to generate that sugar, which is the longer term energy storage. So let's zoom in on the Calvin cycle. There's 3 stages to the Calvin cycle. We're going to talk about them broadly. First is fixation. We're taking 3 CO2s and sticking them together using energy from ATP. In the second phase, called reduction, we are taking high energy electrons from NADPH and generating a 3-carbon molecule called G3P. Okay, you'll notice that this is 3 carbons, and you're going to say glucose is 6. Well, technically the output is not glucose, it's G3P. This will go on, 2 of those together will form a glucose molecule, but this can actually go on to become several other different types of organic molecules. Don't worry about that. For us, just think of it as sugar. Then we have to regenerate this, the RuBP. Okay, the regeneration phase uses ATP energy as well. So we're taking this and regenerating it. Rubisco up here is the key to that fixation stage there. Very important. So we're taking carbons, sticking them together from CO2, generating a sugar. We are reducing that sugar, and then that goes off to become glucose or other things, and we regenerate utilizing ATP. So that's the basic reaction. Really for us, it's realizing that CO2 goes in, it gets fixed together using energy from ATP and NADPH, forms sugar on the backside, but it is cyclical here. So photosynthesis initially did not involve the production of oxygen. We call this early phase of photosynthesis anoxygenic photosynthesis. It took CO2 and some other electron donor. This could be water, it could be hydrogen sulfide, H2, iron sulfide, things like this, and fixed the carbon using those there, using light energy. Now you'll notice no oxygen is produced in this reaction. That is important because life, which is about 4 billion years old, did not start producing oxygen probably until around 2.5, 3 billion years ago. Really, we see like a hump here at 2.5, so we predict that's maybe the start, and then a little bit after that, a little bit younger, 2.4 or something like that, we really start to see oxygen levels increase. So oxygenic photosynthesis took a while to get started. We do see fossil evidence for earlier photosynthetic organisms in the 3.5 billion year range. So our earliest life there was photosynthetic probably, but not producing oxygen. And we don't see plants until much later. So really it was these photosynthetic microbes that changed our atmosphere. We went from very low oxygen to a high enough oxygen level where we could actually sustain large plants and then animals and things like that. So oxygenic photosynthesis, when it started, right, that was utilizing water through chlorophyll to produce sugar, oxygen, and that is the key, right? That's where oxygen started to be produced. Now, there's also been modifications of photosynthesis throughout evolution. There are different types of photosynthesis that occur because we have plants that live in very diverse environments. Things like cacti, you're going to say, they don't have leaves. Well, technically they do. The spines on a cactus are modified leaves. They've evolved to be pointy and no longer have chlorophyll. The stems instead have most of the chlorophyll and the spines are protective, right? So we have problems for organisms that live in hot, dry environments, water loss. Also, this process called photorespiration. So when this reaction occurs, we normally see water being broken down and oxygen being produced. But if the temperature and the amount of water is temperature high, water low, Rubisco can actually run in the opposite direction. Remember, all chemical reactions can go in the opposite direction. So this is called photorespiration. So when it's hot and dry, photorespiration can occur, and we actually take oxygen and spit out CO2, the reverse reaction, right? So we're breaking down our precious sugars there. That's bad news, right? Because we're not generating any energy in this process. It's just spinning its wheels in the wrong direction. So plants have evolved different ways of dealing with this. We've been talking primarily about what we call C3 photosynthesis. It's just your standard capture this CO2 and send it to the Calvin cycle in a mesophyll cell. Here it's noted CBB, it's Calvin-Benson cycle. Sometimes people recognize Benson for this. And then we produce sugars. In different types of photosynthesis, we have C4 and CAM photosynthesis. These types of photosynthesis evolved to deal with the challenges of hot environments and dry environments. We see many grasses doing C4 photosynthesis, corn and things like this. So C4 photosynthesis separates the processes into two different cells. We have the mesophyll cells where we create this C4 compound, and then we're actually really capturing the CO2 and concentrating it in the bundle sheath cells. Okay, I'm having terrible flashbacks to my plant metabolism class in grad school. Basically what's going on here is we're capturing the carbon in this molecule and then storing it in here and just like getting super high concentrations of CO2 to force this to go through, right? Not run backwards and do photorespiration. So here in C4, we're separating out the processes. CAM photosynthesis, on the other hand, separates out the cycles based on time of day. So at night, there's much less heat, right? So water loss is lower. So we see the stomata opening at night in CAM photosynthesis, letting the gas in. They capture that carbon dioxide, that carbon in a C4 compound. They send that over and store it until the daytime where the stomata close. Then they utilize that CO2, that C4 component in the Calvin cycle and make their sugars during the day. So we have separating it out spatially and separating it out temporally or through different times, right? So in CAM photosynthesis, stomata open at night, minimizing water loss, close during the day, again minimizing water loss, and we store that CO2 that was captured at night. Now, that might seem less efficient, and technically it is in terms of like overall growth rate, right? But if you have to live in that environment because there's no competition or very little competition, that's a very successful strategy, right? So we see a lot of these organisms growing very, very slowly over time, particularly the CAM ones where there's limited water. At a broader level, right, we're cycling that carbon. So plants consume carbon dioxide, release oxygen. Other living organisms, plants are living organisms, but other living organisms, non-photosynthetic living organisms really, consume that oxygen and those plants to get the carbon and release carbon dioxide in the process. So this is constantly cycling. But I want to remind you that plants don't really need us. They have evolved to work with us, spreading their fruit and things like that, spreading the seeds. But in this overall reaction, Plants also can break down their own sugars, and they have mitochondria as well, so they don't need us, but we really, really need them, so plants are vital to our survival. Let's finish up with a review question. Which step of the Calvin cycle describes adding carbons from inorganic CO2 molecules? Go and pause the video, think about this. We're capturing carbon from CO2. That is called fixation. So we saw in the previous section that the light dependent produce that ATP and NADPH, but they don't store energy long. We need to utilize that to make sugar. So the Calvin cycle uses that ATP and NADPH to build G3P, half a glucose molecule. It does that first by fixing carbon from CO2. It sticks the carbon molecules together, and then we reduce using electrons from NADPH to build G3P. And then we have regeneration, which rebuilds the RuBP, which can then go on to accept more carbon from CO2. So it's cyclical in nature. The earliest photosynthesis was anoxygenic. So it did not make oxygen. It wasn't until we started having oxygenic photosynthesis that we could start to have other types of organisms that were really much larger and things like that. Photorespiration can happen where photosynthesis effectively runs backwards, wasting energy. This happens in hot, dry environments, and we're taking in oxygen and spitting out CO2, so it's the reverse, and we're not generating any energy in that process. Different forms of photosynthesis have evolved to try to avoid this problem. C4 photosynthesis and CAM photosynthesis. People are studying these intently to help hopefully engineer better crops that can more effectively prevent photorespiration. Energy in the carbon cycle cycles between the autotrophs and the heterotrophs, although I want to remind you, these ones really don't need us that much. We need them, though. We need those autotrophs. All right, that's it for our photosynthesis chapter.